Hello, everyone. I hope you are doing well. This video is regarding the bacterial growth curve, which is very interesting and important topic. I hope you will get the knowledge from this video. Let's start the video. Bacterial growth curve. Bacterial growth curve has four distinct phases, viz. lag phase, log phase, stationary phase and decline phase. A lag phase. The bacteria first undergoes a period of acclimatization when they are inoculated into the sterile nutrient medium. During that time, necessary enzymes and intermediate metabolites are synthesized, and before multiplication the bacterium reaches a critical stage, and finally multiplication takes place at this stage. Hence, this stage is also known as initial stage or dormant stage. The duration of this phase depends on the type of bacteria, quality of culture medium, size of inoculum and some environmental factors like CO2, temperature, pH, etc. The average time of lag phase is 2 hours, but it varies from species to species and ranges between 1 to 4 hours. Log phase. After the lag phase, the next phase is log phase. In this phase, the bacteria undergo cell division and their population increase exponentially at a logarithmic rate. Hence this phase is also known as exponential phase. The number of viable count, when plotted against time, it gives a straight line of inclined fashion. The average time of log phase is 8 hours, but also varies with the bacterial species. Stationary phase. In this phase, the cell division of bacteria is almost ceases due to exhaustion of nutrients and also the accumulation of toxic products. Secondary metabolites like antibiotics are also produced in this phase. The cell death starts at a slow rate and is compensated by the formation of new cell through cell division. The total cell number increases at a slow rate, but the viable count remains almost constant. The duration of this phase varies from few days to few hours. Decline phase. This is the last stage of bacterial growth cycle. In the phase of decline, the total number of cells remains constant, but the number of viable cells gradually decreases due to exhaustion of nutrients and also the accumulation of toxic products. In some cases, a few cells remain viable for a long time. These viable cells probably grow by utilizing nutrients released from dead cells. The cells attain maximum size at the end of lag phase and become smaller in log phase. Isolation methods for pure cultures. A pure culture is usually derived from a mixed culture by transferring a small sample into new, sterile growth medium in such a manner as to disperse the individual cells across the medium surface, or by thinning the sample several times before inoculating the new medium. Hence, pure culture is defined as a laboratory culture containing a single species of organism. Importance of pure culture. Desired known species of microorganisms can grow once pure culture is isolated. Pure culture can be correctly identified for accurate study and testing of microorganism in a clinical environment. Experimenting with pure culture ensures the same results after repetition of experiment many times. In pure culture, the spontaneous mutation rate is low. This slide is showing the flowchart of methods for the isolation of pure culture. There are various methods which can be used for the isolation of pure culture. Streak plate method. The principle of this method is that, by streaking, a dilution gradient is established across the face of the Petri plate where bacterial cells are deposited on the agar surface. This method is used most commonly to isolate pure cultures of bacteria where a small amount of mixed culture is placed on the tip of an inoculation loop and is streaked across the surface of the agar medium by different fashions, and the plates are incubated to allow the growth of colonies, and each colony is the progeny of a single microbial cell, thus representing a clone of pure culture. Finally, the microorganisms are separated from each other. The isolated colonies are picked up separately using sterile inoculating loop and re-streaked onto fresh media to ensure purity. This slide is showing the different ways of streak plate method for the isolation of pure culture. Pour plate method. The main principle of this method is dilution of the inoculum in successive tubes containing liquefied agar medium so as to permit a thorough distribution of bacterial cells within the medium. In this method, the mixed culture of bacteria is diluted directly in the liquid agar medium tube, 42 to 45 degrees Celsius, and mixed well. In this method, fixed amount of inoculum, generally 1 ml, from a broth sample is placed in the center of sterile Petri dish using a sterile pipette. The contents of each tube are poured into separate Petri plates and then allowed to solidify. These plates are incubated at 37 degrees Celsius to develop bacterial colonies in both within the agar medium, subsurface colonies, and on the medium, surface colonies. Finally, isolated the colonies and are picked up by inoculation loop and streaked onto another Petri plate to ensure purity. This method is also used for determining the number of viable bacterial cells present in a culture. This slide is showing the way of pour plate method diagrammatically. It is very clear picture of pour plate method. Spread plate method. The principle of this method involves using a sterilized spreader with a smooth surface made of metal or glass to apply a small amount of bacteria suspended in a solution over a plate that needs to be dry and at room temperature so that the agar can absorb the bacteria more readily. A successful spread plate will have a countable number of isolated bacterial colonies evenly distributed on the plate. The technique is easier to quantify bacteria in a solution. A drop of diluted liquid from each tube is placed on the center of an agar plate and spread evenly over the surface by means of a sterilized bent glass rod. The medium is incubated for the colonies' development on the agar medium plates. The isolated colonies are picked up and transferred onto fresh medium to ensure purity. This slide is showing the spread plate method diagrammatically. This slide is showing the isolated pure culture by spread plate method. Serial dilution method. A microorganism that predominates in a mixed culture can be isolated in pure form by a series of dilutions. The inoculum is subjected to serial dilution in a sterile liquid medium, and a large number of tubes of sterile liquid medium are inoculated with aliquots of each successive dilution. The aim of this dilution is to inoculate a series of tubes with a microbial suspension so dilute that there are some tubes showing growth of only one individual microbe. Example: a culture containing 10 milliliters of liquid medium, containing 1000 microorganisms, i.e., 100 microorganisms per milliliter of the liquid medium. 1 milliliter of this medium is taken out and mixed with 9 ml of fresh sterile liquid medium, then resulted medium have 100 microorganisms in 10 milliliters or 10 microorganisms per milliliter. Further, 1 milliliter of this suspension is added to another 9 milliliters of fresh sterile liquid medium. Each ml is now contained a single microorganism. If this tube showed any microbial growth, there is a very high probability that this growth has resulted from the introduction of a single microorganism in the medium and represents the pure culture of that microorganism. This picture is showing serial dilution method very beautifully. Special methods. 1. Single-cell isolation methods. An individual cell is picked out by this method from the mixed culture and is allowed to grow to procure pure culture. There are two methods used. For the isolation of single cell, I. Capillary pipette method. Several small drops of a suitably diluted culture medium are placed on a sterile glass coverslip by a sterile pipette. Examine then each drop under the microscope until find one microorganism in one drop. This drop is then removed with a sterile capillary pipette to fresh medium for multiplication to yield a pure culture. This image is showing the I. Capillary pipette method. Very clear. E. Micromanipulator method. Micromanipulators permit one to pick out a single cell from a mixed culture. This instrument is used in conjunction with a microscope to pick a single cell, particularly bacterial cell, from a hanging drop preparation. The micromanipulator has micrometer adjustments with micropipette that can move in all directions. A series of hanging drops of a diluted culture are placed on a special sterile coverslip by a micropipette. Hanging drop is searched for a single microorganism cell. This cell is drawn into the micropipette by gentle suction and then transferred to a large drop of sterile medium on another sterile coverslip. When the number of cells increases in that drop, the drop is transferred to a culture tube having suitable medium which yields a pure culture of the required microorganism. Advantage: it can be reasonably sure that the cultures come from a single cell. It can obtain strains within the species. Disadvantage: the equipment is expensive and it requires a skilled operator. 2. Enrichment culture method. This method is used to isolate those microorganisms that are present in relatively small numbers or have slow growth rates in the mixed culture. The enrichment culture strategy provides a specially designed cultural environment by incorporating a specific nutrient in the medium and by modifying the physical conditions of the incubation. The medium of known composition and specific condition of incubation favors the growth of desired microorganisms. This method is unsuitable for the growth of other types of microorganisms. Preservation methods for pure cultures. Preservation is a process to maintain pure culture for extended periods in a viable condition without any genetic changes. Once a pure culture has been obtained from the mixture, the microbes may be maintained in the laboratory over long periods. The most important factor of preservation is to stop microbial growth or at least suppress the growth rate. Objectives of preservation: to maintain isolated pure cultures for longer periods in a viable conditions, to avoid contamination, to restrict genetic changes. 1. Subculturing. Strains are maintained by periodically preparing a fresh culture from the previous stock culture. Culture medium, storage temperature and the time interval at which transfers are made vary with the species. Nutrient agar medium is commonly used where most of the heterotrophs remain viable for several months. 2. Storage in sterile soil. The soil is prepared by mixing enough sand with a rich garden soil that makes friable and easy to handle. A small amount of calcium carbonate may be added and the mixture is transferred to screw-capped tubes or tubes plugged with cotton. The tubes are autoclaved until complete sterility. This soil is widely used in preservation cultures that form and quat spores and quat. To this, thick suspension of spores is added to the sterile soil. Moisture is removed by placing the cap and maintaining reduced pressure over a drying agent. 3. Saline suspension. Sodium chloride in high concentration is frequently an inhibitor of bacterial growth. Bacteria are suspended in 1% salt solution. The tubes are stored at room temperature. Whenever needed, the transfer is made on agar slant. 4. Refrigeration. Pure cultures are also stored at 0 to 4 degrees Celsius either in refrigerators or in cold rooms. This method is applied for short duration, 2 to 3 weeks for bacteria and 3 to 4 months for fungi, because the metabolic activities of the microorganisms are greatly slowed down but not stopped. 5. Paraffin method. This is a simple and most economical method of maintaining pure cultures of bacteria and fungi. In this method, sterile liquid paraffin is poured over the slant slope of culture and stored above at room temperature. The layer of paraffin ensures anaerobic conditions and prevents dehydration of the medium. This condition helps microorganisms or pure culture to remain in a dormant state. Hence, the culture is preserved for several years. This method is also known as agar slant culture covered with oil. 6. Cryopreservation. Cryopreservation, i.e., freezing in liquid nitrogen at -196 degrees Celsius, helps in survival of pure cultures for long storage times. In this method, the microorganisms of culture are rapidly frozen in liquid nitrogen at -196 degrees Celsius in the presence of stabilizing agents, glycerol. It prevents the formation of ice crystals and promotes cell survival. 7. Lyophilization. It is also known as freeze drying. It is the method of removal of frozen solvent by sublimation under vacuum, and unfrozen solvent by desorption. In this method, the culture is rapidly frozen at a very low temperature, -70 degrees Celsius, and then dehydrated by vacuum. Under these conditions, the microbial cells are dehydrated and their metabolic activities are stopped. As a result, the microbes go into dormant state and retain viability for years. Lyophilized or freeze-dried pure cultures and then sealed and stored in the dark at 4 degrees Celsius in refrigerators. I request to you, please subscribe this channel and like and share this video to get more videos and updates. Thank you.